Welcome to the webinar Automated AI-Supported Analysis in Alzheimer's Disease Research. My background is neuropathology. I'm working in neuropathology doing general diagnostics on brain tumors and muscles, but also on the field of research. We are working on Alzheimer's disease and develop new treatment, and we have found some interesting things that we actually use now for treatment also in patient studies. But for doing this research, it's very important to be able to quantify and to describe changes in the mouse models that you use. The mouse models are not perfect, but you have to be able to quantify changes if you treat them. So as I've been showing, you can have a look on our homepage, and on the homepage you can find the recent publication, which describes a little bit in more detail about the stuff that I will talk today. The publication is about using AI and the software and the learning mechanisms for segregating A-beta amyloid, for measuring this, and also microglia. But you can use it for anything else. So I will show you some things that you can do. We actually start from scratch to show you how this works, how easy it is, even if a person cannot program anything. It doesn't matter. So what do? We start the software. And what you get is you get this kind of start view where you can have a look for the determination of something. So we are mostly interested in not just in cells, but in objects. So I go to the objects here, and I will open a project that we do. We name this webinar 1. We go in there. Oops, there's a project still in. That's not the problem. Just have a look. Perhaps I used it before. I will just get rid of everything that is in there and start again. So no problem. Webinar 1. Select the folder. So we open a new project. And that's very simple. You have a new project. Then we add in the project slides that we are interested in. So we are going here and having a look at slides. I have some example slides here, slides for testing, and I can just go for amyloid detection. I use these 2 slides here and add the slides to my project. And now you can see what I have here. Here on the top, you see the slides, the 2 slides that I added. Here you have a magnification view on the right of the slides, and here you can go in and scroll in and out with your mouse in the slide what is scanned. These are mice that have been treated with drugs to prevent the formation of amyloid. And then we took one hemisphere always for biochemical assessment, and the other hemisphere we use for histological assessment. And these slides have been stained with the 4G8 antibody against beta amyloid, the central region, and stained with a machine that is called BondMax. And then you nicely see those deposits here in the brain of those mice. In between, you see all these nice neurons here in the cortex. So we have here the cortex region. We have here the hippocampus. We have here the central basal ganglia. Here we have the CSF room with some plexus here. And if you go further down, you may have other locations. So all the mice, we have 2 sides of the mouse hemisphere always on the slide, and then we put 2 cuts on it. So we have 4 micrometer in between those. So we are interested in to measure this. So we do want to know, here's a vessel that is nicely going from the surface of the brain into the deep. So it's very rare that you measure, that you actually can see this. So what we want to do here is we want to measure and compare the amount, so the number of plaques that we have, and also the size of the amyloid plaques. Because if you treat animals, it could be that the number is reduced, but it could also be that the size is reduced. And also you see here already differences. You see plaques that have a dense core here in the middle, and you see also plaques that are more diffuse. So if you are interested in this, we can also segregate for this. So in the first, we want to start to say, okay, we need something that is background. Here on the left, you see this. And we want to have a category, and our category 2 are the plaques. So we start with measuring plaques. I just want to show how this goes. So I go in here and have a look for a location that I like. Then I draw for the machine a learning model, so they can now detect this is one of my plaques. So this is in the violet, so I take another one. I have to give some examples to the software what I actually want to have. It is very important that you do it properly because this is an example. It should be a perfect example for the machine for learning. It will give you later some other possibilities where you can choose always the best. So I choose a few. I need a few to give some examples. We have one here. That's also very nice. So we go for this here. In the start, it takes a few seconds to do this. Also, we can use a smaller one here and go around. And you see it like this and the bigger one. So usually the 5 to 10 examples I take in the start. So then we have this here. And then, of course, I need to have some background because the machine needs to know the background. So I go here. I draw in the background some here. This is background. This is background. And perhaps this is background here. Or then I have some background here as well. This is background. So that the machine can learn what I'm actually not interested in. I could also measure plaques and the neurons in one go. So I could do 9 different categories here. Would be no problem, but just for the purposes now, I just choose a few. I always choose some borders, then some background as well here. So, and then we can have a look whether the machine started to learn when we go on graphs. So it will start soon to give us a graph here, and then we know the machine starts learning. So we can have a look for some more plaques here very nicely. Zoom in. We go on plaques, draw, and go this way around. So it's quite simple. We have a tool. Before we use this software here, we had a tool which we programmed using the Axovision software from Zeiss, which was segregating gray channels, which is actually quite complicated because it takes a lot of time to do this. So that's the graph. So the graph is working. We see the software in the back is learning. We have 3 epochs. And now we can start with something that I like very much here, because I just take this away. I don't need this. So I can go to another place, perhaps here or here, and then I can go in and start the detection mode already very early in learning. So I run the algorithm parallel to the learning that is progressing to see after a few, now we have epoch 5, how does it look like actually. So the software now in the back, we have a grid, a computer group of NVIDIA GPUs that works here. And in the field of view that we have actually now, the machine tries to detect according to what it has learned already in the few minutes now, when I annotated the positive and the negative examples, so the background and the plaques, is annotating now this field here, is analyzing this field, and usually it takes a few seconds, half a minute to a minute, until it starts detecting in this field the plaques. And it will show me everything that it thinks is a plaque. You will soon see this. So the good thing is, when you run this in between, you can control for new objects that you want to add for the learning. And you can also run the learning overnight, but then you have no control in between if you want to learn overnight, but it's not necessary actually because it's quite fast, and usually I stop it after a while when I see that it's working perfectly. So now we have seen the detection here. This is so our test. I can press a button if I like it, then it will be included in our learning. And you see already now, after these few epochs, it has detected plaques and also diffuse deposits in a perfect way. So it is already perfect here. You see, I can add this big one, this big one. I have another big one here, this one, and this one, and this one. And it is already We have not been working long here. It is 15 minutes, including the introduction, and we already have a tool that is actually detecting plaques in a perfect way already at the first run. So we have 8 epochs now. Usually I let it run till 30, 25, 30 if I want, and then I can do the detection. So what you see here now is very easy. I go to another place. I said, okay, good. Let's check this here again, whether it's working properly or not. And I just run it in this field. Now you see the bar underneath. It's running, and we will see what is detecting. And if you want to do more, you can just use later, not just one slide, but several slides. Lily asked SVS. What is SVS? What do you mean with SVS?
It's a format when you scan slides.
You can use any format.
Okay.
We have the Mirax format. You can use import any slides. That's no problem.
Okay, thanks.
It's just independent of the scanner, so
okay.
That is absolutely no trouble.
No, it's a type of document.
Yes,
like,
yes.
Yeah.
You can use Hamamatsu format. You can use the Mirax format, the panoramic format, Aperio. So that's absolutely a problem. If you have any format that you don't know whether, then it's no problem. We can introduce this. So now you see already the plaques are detected, and I take some more, and I can use these also. It's already nearly perfect, actually. So it detects the plaques you see here in a very excellent way, and I let it go a little bit further with some new examples. But here it's already here is something I don't like. You see this here. So I can press this and make it the background because I don't want to have the intracellular deposits. Also, this here is something that I don't like. So, but I could also select just for those deposits in the neurons. That's also possible. So then we can have a look here. I go on the graph, so it learns more. When you add new examples, it goes a little bit up in the precision. So the train goes up, but then it goes down again. So when it goes asymptotic versus 0, you see that it incorporated all the new examples into the learning, and it's continuing with this. So, and now we have in parallel the detection and the learning, and what I could here, training in action. So it trains itself, it goes on, but I could also go to settings here and actually stop and freeze the status, and then it will be much easier to detect. We can do this because I think it's quite good already. So I stop, I freeze it, I apply, and then I go to my viewer and take the other slide. Go here. This is the other slide that I never used before. And I can choose for my report a region of interest. And what we are usually doing is that we use the cortex area we are interested in. It automatically detects the tissue, so you don't have to go close by. So we are interested in the cortex here. I go here, 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 and here. I put this. Then in the settings, I want to have all the raw data. So I save this. I want to have all plaque data. And then I have this, and then I can just press on report. And then the system works. And it starts to detect. So, and it starts line-wise going up and down. You will see in between already the results. So I can zoom a little bit in so that you can see this, how it works from one side to the other, and detects this. We also have a batch mode. So if you have annotated a number of slides that you would like to analyze, so it can be done in a batch mode where you add all your slides, you annotate what you want to have. In future, we are interested in to do this automatically using the mouse atlas, for example, and then saying, okay, you want to have, for example, just the hippocampus region, and it automatically will choose the region. So now we have a look here, what it's doing. So it detects now here all the deposits here. We have deposits here. We have deposit here, some larger one that is double. So we have to learn it a little bit more, but just for today, for the webinar, We just let it run now once to have a look what it actually detects already. Teaching is stopped. It just goes now and tries to detect. And you see, it's not very complicated in teaching. So you have just to choose your right plaques. You can choose any object that you want. It might be in red if you have stained it red. It might be in blue if you have no stain at all, just the Himalayan stain in the background. And then it detects the plaques here. You see nicely it goes to the plaques, also the smaller parts. So we have to teach a little bit more to get rid of these smaller things. But we already see that it is quite fast in detecting some stuff. So that's what I usually do. Then I check how does it work, and I discover some things that I don't like very much. So I can incorporate these in learning. I stop this now here, and then I can go to the settings and run the teaching further. And then I go here and incorporate the right ones for the teaching. That's a big one that I don't like because it's double, so I take this away. Usually that's the easiest way to delete this. So like this, and only choosing the ones that we are really keen on and we think this is perfect. I'll go on here. Here's a very big one. It's not, don't take this one, 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 take this one. This is some diffuse deposit. Here's the question, do we want to have this or not? It will be detected anyway, and we can go here, here, and here. So that's very easy and very simple. And in a second, we will start again the detection on the slide by choosing a new field of new, just to have a look here, you see. So that's actually not very complicated. And for us, it's very interesting when we treat the animals to get an information about the number of amyloid plaques in the cortex of those animals, but also the size. So the size is sometimes very interesting, and sometimes also even the structure of the plaques. Some of the plaques have these core things, and some are dense core, and some are the diffuse plaques here. And it is really depending on the treatment you do that you end up with plaques that change their appearance in a way. So let's have a look for the graphs. We have added some of the new objects here for learning, so it goes up a little bit. And now we can keep it a little bit running just to go for this. I leave it a little bit in the background for a few minutes. So we will come back to it. It's learning now in the background. I will just show you the paper that we actually did, some information about this. In the paper, we compared two methods using machine learning and using the method by size, which is a program. It's called AxioVision, which is an excellent program which you can use very nicely, but it's not learning. So this program is a system where you can segregate pictures according to grayscale, colors, for example, and finally you end up with pictures that are black and white that you measure. And you can determine plaques, you can determine microglia. So we programmed this. The programming with such a tool, it takes already very long. So approximately 2 to 3 days you need for programming. Here in the AI, you have seen it's very fast. And what we have done here, here's an example. We have detected amyloid plaques, diffuse and dense core plaques in different categories. So we have the smaller category here in green and the bigger category in red. And we also taught the system to detect microglia, only microglia that are activated and have a cellular body, not the small processes. You can also run the process detection if you want. So you can do detect what you are actually interested in. We compared these methods because we wanted to know what is actually better. And we have seen that with the AI, if you do a plaque programming, depending on what you want to do, it can take up to 4 hours, but we have seen now we are not even taking 15 minutes and the detection is quite nice already. When you run the detection, With the AxioVision, it takes an hour to 1.5 hours. You have to clean up all the bio things. It's quite laborious, and you have to work with it. In the studio, it takes about 10 minutes for one slide. For the microglia, it's even more work. For the detection, it's up to 2 hours if we work with this, but it's also about 10 minutes in the studio. So it helps a lot to save a lot of time in the start for programming, but also later in the plus. And here we have similar pictures as you see here. We have compared this and also the correlation. So the software gives us the same comparable results with respect to plaques. So this is the same slides measured with different systems to be sure that everything works fine, and it actually works really nicely. And we have a tool where we can segregate small plaques, medium-sized plaques, and large plaques. And also here we have a very nice correlation between those. It's very interesting. The AI is much better in detecting small plaques because it is more precise in a way, whereas the AxioVision software has problems with the background and with the plaques. That depends a little bit on the programming, but in the end it is like this. We compared this also with ELISA data, so we measured the mouse brains with ELISA to see what's ongoing. And the same is true also for the microglia detection, which we will run in a second. So you can download this, you can read this, also the discussion and some information about the background, what we did. So let's go back, and we are having a look at the graph. So we are at epoch 18 approximately now, and you see it goes down. We have 3 times or 4 times added new examples, and it's learning like this. So now we can go into our viewer and select another location of interest for us. In parallel, we let it going, so we mark the location, goes around here, take a piece of the cortex, so, and then we run the report, and then the software starts to use. In parallel, we are not training now, so it depends on the machine you have, what is better. You can go in parallel with training, but sometimes you have to stop it if it's very complicated. There was a question about the paper. Yes, the paper is as PDF on the homepage, so you can have a look. And there was another questions. When you run the model, you have to check in between. So it can be that it is overlearning and it's getting worse. Depending on the detection you do, you get an experience after a certain time whether it's good enough or not. So this is something that is a bit of complicated for machine learning to stop itself. We have a mode of validation here. You see this down here. You can use this validation mode. You mark a test area and you mark all the subjects that are positive objects, and then you can run the validation mode, and it will run on top of this detected and marked objects, and then you can actually see how accurate it is. So that works quite nicely. So the paper is on our homepage, yes. So it is also soon, it's a preprint version we have now. It is soon in the corrected version. I just got the PDF actually today from Journal of Alzheimer's Disease, and I sent the corrections back, so they made some errors, but that is usual. So you see here now how it's detecting. So it is detecting all the bigger ones, the smaller plaques, and so on and so on. And this already after 20 minutes. So my suggestion is always to run it or leave it run at least an hour or two, so that you reach epoch 20 to 30, 35. If you want to teach it longer, you can run it longer to 100. Not more than 200. After 200, it does not seem to help much. It might just get a bit worse. If it in between...
I think that there are two questions that maybe we can address. One question is, how do you prevent overfitting of the model? So I think that I can maybe start with an answer, and preventing overfitting of the model is really dependent on the data that you are using to actually train the models. So you really need to make sure that your data is representative and diversified, and that you are not just sticking to single type of a use case. So variety of data is very important. And I don't know, yes, do you want to add anything to that?
No, I'm always working as long as I see that most of the background problem is solved. Here you see, for example, in bluish, there was something added, I don't know why, which is background here as well. So I just declare this as background, and then the machine learns this. And also we have another example here, this one here. This is actually a leptomanageable vessel that is positive. This is CAA. So I declared this now as not a plaque, so as a background. But you see here, if you go a little bit further, how it detects is very nice. Here we have something that we don't want to have. We declared as background. But here, all these detections are perfect. So you go here a little bit, here small ones, here small ones. This is not very nice here, so I declare this part as background first. Now it's learning. So I can take this away, and then I declare this as background, just to prevent that it is taken into more consideration later. So, but it looks already very fine. So to me, it is becoming much, much better. And you see here, all the small things are detected. It goes quite nicely. You can use fluorescent images. That's even easier. That's absolutely no problem. You can scan fluorescent images with a Kian scanner, but you can also use the Mirac scanner. That's absolutely no problem. Whether you have fluorescence or normal images, it's absolutely no trouble. Fluorescent images are even easier because usually they have a really black background if they're done properly. But you could also segregate multiple colors at once. That also works very nicely. Depending on your scanner, what the scanner actually delivers. Here you see I put this as background, and so on and so on. So that is now actually running through this slide, and when it's finished, I will show you what the result is. You can get an Excel sheet. In the Excel sheet, you have each of the objects that have been detected labeled with the location, also with the size, and you can use this then in other programs if you want to use it for diagrams. You can also use the objects with the diameter and the location to draw new images and paste them in. That's also possible. So anything you want to do is possible with this. So there's usually no problem. Here we have some double counting. We have one plaque here, and then the bigger one here. So we have to have a look how it goes on. So it looks already very nice. And if we have a look for the teaching, I have added some more here, also some more background. It goes up again. So you see it is quite efficient. So let's wait until the final parts are detected here, and then we can have a look at the Excel sheet and how it looks like and what the data, how the data are presented. So this is actually all. You have not to do anything more. You just go in, you annotate your cells, you annotate your objects, whatever you want to have. You start the learning mode, it's training, and then you have a look in between a few times whether the algorithm gives you a good response on what you have put in as objects and background. And as soon as it's ready, everything is done, then you stop the learning and you open a new project and continue with the next. And then you can also load more slides here to analyze all the slides, and you just have to mark the region of interest that you are looking at. In future, this will become much more easier. As I said before, we are working in establishing a combination of a mouse, Paxinus mouse, atlas, and automatic detection in mice of cortex, for example, or other structures, so that the AI can first detect the location you want to have, and then the objects in the location. And if you run this fully automated, then you don't have to do anything anymore. You just learn the objects, and then you will get the segregated information in your results Excel sheet with all the objects. So now we are nearly done.
There is another question, Jens.
Yes, I will have a look.
Okay.
If you want, yes, you can also use images. This is another mode. This is the cell mode. It will detect different images and give you also a gallery of images. This is here, but for the plaques, I use another mode. You see also the epochs for teaching. So you can do this for single cells, for example, if you have cytology, or if you have cells within a tumor, special cells that you are looking at. So you can also teach it for certain cells. That's also possible. So just for this now, I would like to show you how does it look like with the results. So you can go into, we call this webinar 1. Here is the Excel sheet. 2028 is our time here in Oslo. And let's have a look. So that's how it looks like. Here we have now a single sheet where we only get the amount. We can also segregate it in small. So we have the objects, 205 objects, the area, object, and so on. And then we have the raw information for each of the objects. For example, the area here. So this is what we now wanted to have. It is also possible to export a diameter or whatever you want. It doesn't matter. So the geometrical data is in the software, and this can be, so to say, exported as it is wished. This is absolutely possible. For us, it is important to have the area and what it is. The category, if it's something else, the category would have another name. Okay, so this is this, and I will put this in a new, just make a new folder. We call this webinar 1 - 2, perhaps, for the next. I already make a folder. So this is now finished. I have the area done. I could now measure all these slides that I have imported. I can also stop the teaching now and freeze the teaching, and everything is fine. And here I can have a look at the epochs 26. It goes a bit up here. So that's what it is. So it's actually done now. And I could analyze a lot of other slides. If I have slides that are stained differently, where the stain is a bit weaker or a bit more, I can start from scratch, or I can use this learning and teach it a little bit differently. So I will just save this project, and then I will.
Can I ask a question?
Yes, you can ask a question.
Thank you. So once you've done this teaching, or the machine learning, what you need, can you after that just basically select this whole half, section?
Yes. You can select.
And say now analyze this whole thing.
Yes. That's what I just did. You can now load in a lot of slides. In each slide, you choose the region that you want to have measured, and it will automatically measure. And the next is what you have here. You have this was the folder, and here you have the checkpoints. This here is actually the teaching. So if you use this file, you can use it, reuse it as often as you want. This is your teaching. This is actually the brain of your analysis now here, the checkpoints. So you can just copy paste it into another project and use your learning for another project, and you don't have to reteach this. So it's very simple. So now we start the software again. I always close everything before I do a new thing. So we can use go also for an object again, and then we go in for NRL. I use the seminar number 2, so select the folder. And now I can choose a slide. Let's have a look. I do something that I never did before. I told this to Chen. She does not need to be afraid because
yeah, I was a little bit hesitant about trying something for the first time during a live webinar, but Jens is a very courageous person, and we decided that we flow with him.
Yes, I just have to have a look where I have the slides for this. So this is not this one. I'm looking plugs for testing. No. Oh, wait. It's in here. So we go in here. We have it here. Here we have the slides. That's where I'm. So we do. I wanted to try something new, and I will use this here actually. These are 3 slides where I have a new end stain. So here you see a very nice stain of all the neurons in the cortex and all the neurons in the hippocampus, for example. So this is new N, staining all the neurons. And I would be interested in to detect all those neurons. So let's have a look how it works. So we have the background, and we can call this a neuron, for example, the more weaker one. These are in another, where is this? Oh, an N, neuron weak. First, my N is not working. What is with my N? Oh, I have a problem with my keyboard here. That's funny. Why is the N not working? Okay, call it neuron. We W, and then we put another. We call this. There's my neuron dark. So weak and dark. I don't know why the N is not working. It's a problem. Doesn't matter. So now we use some the darker one. We go on this one, and I will teach the software. Let's say for this dark neuron here. That's one. Number 2. This has anatomically nothing to say now. I just choose 2 different objects just to show you how it works. These are all the same neurons. They are just in a different layer depending on the cutting. That's why they are dark or not so dark. So we have this. We also take the dark one here. So now we have 6 objects. Now we take the little bit lighter ones. Let's take one of those here. They appear with a different color then, violet or green. And I, of course, can add other objects here, like cells that are not stained, that are not here, like this here. So we have 5 here as well. We'll take some more. It's important that you take care of doing this properly, because the worse you choose, the worse will the learning be. So these objects are quite important to choose properly. So then we can go for some background. We always have to tell the background. This is background, this is background, this is background. It's very important that you choose proper examples of the background. The more, the better it is. It's not so difficult, just to give the software really a possibility to segregate from all the other things. Let's have a look what we find here. Some background. So some background here as well. Oh, here's some very nice dark neurons. We can use some more of these here. And so on and so on. So let's have a look. We go on the graph. It's not started yet. Perhaps we need some more darker ones. We'll have a look. So when it's sufficient, it just starts the learning. But we need some good examples always. That's very important. These are very dark. And it's important that you choose objects that yourself can segregate with your eyes. If you choose similar things, the software has the same problem as you have, and then it doesn't make sense to show some weaker ones. Go for this one here as well. So, but you see, you don't have to program anything. You just actually go through the slide and choose objects that you think are representative for what you want to measure. And either you choose the round one, or you have something like I showed you with the microglia, or you can also choose just the big neurons here in between if you want, or everything. So it's started learning here now, epoch 3. So it goes up and down because we add more things. I run it now in full resolution mode. You can also run it in a resolution mode for the plaques, for example. If you have bigger objects where you don't need full resolution, then it's even faster. But here, for the sake to show you how exact it actually is, I just use these kind of things. So all these oligodendrocytes here in the white matter. This is a vessel stained here. This is here ependymal cells. We don't want to have these. It's all background. So that's what I usually do in between. When it learns, I add some background here and there so that it has sufficient amount of background. So that's how it looks like. Oops, that's a bit too big. We don't need this. You leave it, it's no problem. But I usually choose background objects in the similar size as the objects that I'm looking for. So this is this. And now we have, here you see the location that we chose here and here and here. And now we can go somewhere else, perhaps here in the cortex. That's not very fine from the scanning. We look for something that is more neat, something like this here. So let's have a look. 6 epochs is quite good. We could try and have a look what is going on there. So I just start this and have a look what type of objects the software actually is detecting now. We have some background here with the oligodendrocyte. We have some neurons here that are a bit darker, some a little brighter. So let's have a look whether the learning gives us some reasonable results. And what I do is I run this algorithm, and then I select always the positively detected perfect things, add these for the learning. Just by pressing on the right mouse click, I just add these to the learning, and all the others I leave, and I may also add some more background. Is the machine looking for the same color? Can it search based on shape? It is all together. It is not just color. This is the point. It is color. It is distribution of color. It is shape, is everything. And you see this here. So, I mean, I did not do anything. It has 6 epochs learning. And what you get is here. This is a background. We don't want to have this. This is one that is soft. This is a darker one, a darker one, a darker one, a darker one. This is a light one, light one, light one. I would like this to have a darker one. This is a bright one. This is a bright one. This is okay. This is okay. This is okay. So this is also a dark one. This is a dark one. This is a dark one. Light, 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 light. You see, it is no problem at all to get these things detected. Very easy. I did not do anything else. So this is not a trick. This software, we never used this slide before. Chen said, oh, don't do this. No problem. I am fully agreeing that this software is really working perfectly. It's absolutely no trouble. So here we have cortex again. And we are now in the teaching. So we added new examples, 8 epochs. Now it goes down again. So it has incorporated the new examples. And now I just run the algorithm again on this new part. And immediately it uses the information from the teaching to detect now our objects that we have segregated. So I would expect some of the darker ones will be in the dark group, some of the brighter ones will be in the bright group. it doesn't make sense yet now, actually, for some research purposes. It's just to show you that it can detect just different things. It could be different neurons as well, if you have different colors, for example, or different sizes, different shape, anything. So it can be anything. The software works detecting on size, on shape, on color distribution, everything. Yeah. So, Chen, the question is answered.
Yes, thank you.
Yeah. Any other question in between from one of the audience?
Yeah. So actually, you would like to differentiate between the darker and the brighter.
That was the idea now. Yes, that was precisely now the idea, because the darker neurons could be something special depending on the stain, and the lighter neurons could be another a group of neurons. So I would be interested in, is there a certain distribution of darker and less dark neurons in this cortex area? For example, if you treat a mouse or you do anything else and you want to see, has it an effect? To measure this with a general software now that you can get, it is quite complicated. So you need some software that is intelligent to do this. And it will work a little bit. It takes a while because it's a lot of objects here now in this location. But suddenly it will all appear at once. So at first it looks like, oh, what is going on here? But you have this green bar, so it helps you that you see, okay, the software is still working. It's not stopping. And since it's now teaching itself in the background and in parallel detecting, it takes half of the computational capacity for one of these two. So one has to be a little bit careful to wait. And that's it. If you do this by hand or you do this with another software, it will take overnight or even longer. So you sit really long to do this. You have to paint each of the neurons, and then it will be put in the categories and everything. So it takes quite a while to do this assessment. And here you just have to teach it. Then you say this is the region of interest, and then it will go through this region of interest and detect all the stuff you have in the cortex here. So let's have a look. We have to wait a little bit.
Yeah, and by the way, is the brown staining? Is it the same the same staining that is used for the beta amyloid plaques? Is it because it's both brown?
This is the same color, but the primary antibody is different. It is the same coloring kit, so to say. The difference is that the primary detection antibody is different, but it's the same brown. But you could use also green, or we also have red color, for example, when we go for. So precisely. So now we can go in.
Like you said, everything appeared at once. Okay.
Yes. In this mode for algorithm, everything appears at once, and it appears actually all what was in my field of vision. So let's have a look. Now we have a look. How exact is this? So these are the dark ones. It looks good. These are the more brighter ones. It also looks good. So I see it. It works actually very nice. You see darker ones, brighter ones. So here are darker ones. Here's a brighter one that is violet here as well. So if you are not satisfied with this, you could now choose more examples to teach the system even more. You just press here with the right mouse and choose more of the darker, for example, or you choose also more of the brighter. Here's one that is bright. So, and by doing so, it will be added to the teaching. So it's absolutely no, here is a brighter one, for example. So it doesn't make sense now to have it, but it shows you actually that you see the weaker stained, or those neurons that are not in the same layer are detected in violet here now. So you have seen how fast this was. How long did it take? 10 minutes altogether. And here go a little bit more down here. So I think this example is finished. So you can use this now. And if you want, let's have a look at the graph here. It's now 12 epochs. You could stop the learning, but you could also now do the following. you take your new slide, you take another slide, take this one here, for example. Now we choose the location of interest. We mark our, I'm interested in this part here. For example, this is the special cortex location, and I would be interested in to do this analysis here. And then you press on report. And in the report, it will give you, then let's have a look how long it takes. So we could accelerate it a little bit by freezing the teaching, and then it's a little bit faster. Then we have full capacity of the GPU. So now it's running, and in the report mode, it is scanning like this. So you will see some results on this side here, and then more and more results. What? Start again. The freezing stopped it. So now we started. We can stop now. It's 20:56. How long it takes until it? So that's why the bar shows you whether it's running or not.
Yeah. Can you can you in the meantime maybe say something about the compute power that you use to?
Now we have a virtual machine here. So what you see is a screen of a virtual machine. That's why I have the background AI machine, so that I know that's not my computer. I am connected to a virtual machine that is in our lab, and this virtual machine has a GPU from NVIDIA. This is a grid GPU that we use for the computational power, and this virtual machine is using 4 Xenon processors with 3 gigabit for general computational power, so it's quite good. And yes, and we use it especially just for 2 analyses. One analyze is the Studio, and then we have another analyze. This is this software here. The ACO Vision software is using it for mouse tracking when we do water maze experiments, for example. So here you see how it works. It started to analyze here already in this location, and as soon as it detects more cells will be added, and it's excellent. It works really nice. I like it very much. So I can tell my lab tomorrow that they can use it for the neuron detection as well without trouble. So it works immediately. So you see this, if I go in, you see this already here very nicely. It really detects all the full neurons. Here are some smaller things. Perhaps we have to, here's something that is missing, but that can be changed when you teach it a little bit longer. So that was just 10 minutes. So I would run it half a day or a few hours, and then the teaching becomes much, much better, much more exact, and all the small errors that are still available now will be then vanishing without any trouble. So you see already in the start how exact it detects depending on your primary selection.
Jens, I think that we are approaching the end of the hour, and I know that some of the people already typed that they have to leave for another meeting. So I think that maybe we can
answer questions if somebody wants to have another question.
Exactly. So let's get some more questions, and just to let you know that we will also have this recording available in the coming few days, so you can visit it, and we'll be very happy to take any questions also offline. But if you have any questions now,
just want to know. Yeah, just want to know how to access actually this program. It seems very nice. How do we get access to this to do our own analysis?
Yeah, so I will. I think that I have your email when you registered, so I can follow up. So I will follow up with an email, and we will be happy to discuss. Absolutely.
Thank you very much. Well, thank you both. My apologies, I have to leave. Thank you.
Thank you very much. Have a nice day.
You're welcome.
It was very great. Thank you.
Thanks. Okay.
So we can wait until this is done.
Yes.
So we have the plaque detection, we have the neuron detection, and now it's running and detecting all the neurons. It's really, I like it. It's really good here. So it's.
It's. I have to say that I was really a little bit I was a little bit worried because usually when you try things for the first time on live demonstrations, something tends not to work, but you seem to master it. So
that's good. This works. I mean, the studio is really good, so we can try even such things. So it's a live presentation. It's not pre-recorded
yes,
or. I think I always prefer if I see something that is not prepared fully. If somebody presents this to me in the lab, I'm always very happy if I can take part also in a little bit of a discovery. So like here.
Yeah.
So you see, it takes a bit of time, but I mean, what did we do? We had now 1 hour seminar detecting plaques quite perfectly. So I would wait a little bit more for the teaching of the plaques, just for security reasons. The same here with the neurons. So we taught the neurons, and we're now detecting the neurons in different colors, more darker ones and more brighter ones. So we can also use it according to the shape. Small and larger neurons is also possible. And this is all within 1 hour. You establish 2 detection modes that run nearly remotely in the background now. So I think it's really efficient, very nice and precise. And the most important is it's really easy to use. It's not that you have to have special skills in programming, nothing. You don't need anything. You just have to know what type of object is your object, and does this object that you are interested in is different from the background, different from other objects. So if you, by your eye, can detect something, you can teach the system, yes, this is my region or my object of interest. So absolutely no trouble. It is you don't have to be a pathologist, you don't have to be a specialist in morphology. If you use your healthy eye, you can choose the objects that you are interested in to automatically detect them and give you a quantification, size, location, everything. So, and this is, of course, the detection. If you have more objects, takes a little bit longer. But what you can do is you can run this then remotely in batch mode overnight. Let's say you have 10 slides from 10 animals. You just tell the region of interest, and then you start the analysis, and you come back the next morning and you have the results, and you just have to invest half an hour for teaching. Perfect. You come back after half an hour and let it run another half an hour. Then you start it overnight, and next morning you come back, you have the analysis of this. So it's really cool. And if your staining is done in a proper way, so the quality of the staining is good, then the results will be good. Is the staining not as good, then you can teach the system also. It doesn't matter. And it will give you results, but you may have to think about whether all the objects that you detect have the right color or not. So you have to perhaps group something together, brighter and darker stained things, if this is in a single group. So it depends really. But what we also did is sometimes we have some problems with the staining. If you use a new antibody, it's very dark. And if you use an antibody that was coming from the fridge after half the year, it might be a bit weaker. You can just adapt your detection to this weaker stain without any problem. It's very easy, and then you can run the detection again over the same slides, and you will get a fine result. So that is actually absolutely no problem. And you do not have to rely on other people giving you an analytic tool. You can do the analysis from scratch by yourself. So it's not that you are dependent on anybody else. You can choose what you want. So now the detection is nearly over here. You see now everything is detected. The green bar is gone. And now we have all the objects in here that we have been detecting. And now we can have a look. Let's see where it is. I go into the where we now want to. So we have this here. It now calls it different. We have the neurons weak, we have the neurons dark. So we have 900 neurons, nearly 1,000 neurons of those. Here, objects per category again. So if you have a single slide, then you have only double the same. But if you have more slides, you get all together and by slide. And you can also get the information per object. If I click in the software that I want to export each of the raw data outputs, and then I get also the raw data for each of the objects. So that is working very nicely. I just freeze this down, apply, and that's it. So I think we are done for this. If you have more interest, we can also do something else. Officially, the time is over for 1 hour.
Thank you very much for joining the webinar.